overview of the properties of viruses and general virology. And so on this first slide, you're looking at the structural diversity primarily, and you can see some pictures of that here with these bacterial viruses. In this particular case, this is a phage that primarily infects E. coli, and it has a very frightening and very human-made appearance. Here's a sort of a rod-shaped looking virus that is actually more like an ear of corn if you looked at it more closely. And then here's some rabies viruses which look like little bullets, and then just a more typical virus, like this guy here, like you might see the SARS coronavirus looking like something more like that. As a general rule, they're all obligate intracellular pathogens. They can infect both prokaryotic and eukaryotic cells, and so obviously prokaryotic viruses are referred to as phage, and they often look like this. They have a capsid and then a tail, and then many of them have tail fibers which allow them to bind to bacterial surface receptors. And then, of course, the eukaryotic viruses we just refer to as virus. We definitely don't call them phage. They all lack energy generating or biosynthetic machinery, so there's nothing packed in here that would allow this thing to self-replicate. It has to inject its genetic information into a susceptible host cell in order to complete its mission. The viral genome directs host cell replication machinery to make more virions, and that's a general truth. This genome has the blueprint essentially for all the polymerases and virus parts. But and the virus obviously needs the host cell in order to provide ribosomes, for example, for translation and sometimes polymerases even. But many times the viruses bring their own polymerases, so they're doing a little bit of the work. The life cycles of viruses can be lytic or lysogenic, and I'll show you some images of that later on. But as the name suggests, a lytic virus is one that actually will result in cell lysis, whereas a lysogenic virus typically inserts its genetic information into the host chromosome and stably replicates along with it. The genomes can be both DNA or RNA, and I'll show you that as well. Just a little bit about taxonomy, which is my least favorite aspect of biology. So in virus taxonomy, it's quite a bit different than bacterial taxonomy, and so you can see with this, with human herpes simplex virus, the species name is human herpes simplex virus, as opposed to E. coli, where the species name is coli. These guys have a very long descriptive title. The genus name simplex virus, and so forth. And so again, it's a different naming system, but I'm not so concerned that you know that. There is a classical classification system that's based on the nature of the nucleic acid in the virion. So DNA or RNA, single-stranded RNA versus double-stranded RNA, even polarity of the RNA, whether it's plus or minus RNA, and so forth. The symmetry of the protein shell is also important, or capsid in this particular case. So the symmetry can be helical or icosahedral. So there's a few different arrangements that allow you to distinguish phenotypically. The presence or absence of an envelope is also important. There's envelope viruses and non-envelope viruses, and then the dimensions, how big the thing is. Couple aspects of virus pathogenesis. One thing that they can do is directly cause cell damage resulting from their replication. Just the mere fact that they're taking over the host cell machinery and stealing ribosomes and commandeering the membrane trafficking or the internal endocytic trafficking systems can actually kill the cells and cause lysis in and of itself. Sometimes it causes cell fusion, meaning that the cells actually kind of stick together, which is not always ideal. Damage can also result from immune activation or suppression. So suppression, obviously, with like HIV, which we'll talk about, but also immune activation, which, again, we're seeing with SARS-CoV-2. Immune activation is a huge part of the reason why we're having such high mortality in the elderly. And one of the other more startling things that viruses can do is cause cell transformation. And transformation is just a polite way of saying cancer, And so cell transformation can be caused by lysogenic viruses that insert their genetic information into the host genome, and this can actually cause disruptions in the cell cycle. And the net result can be tumors and cancer. So the Baltimore classification scheme is the sort of modern approach for classifying viruses based on the genetic material that's present and the replication strategies. Group 1 viruses tend to have genomes that are more similar, and genomes and replication mechanisms that are more similar to us, the humans. And animals. And so they have double-stranded DNA genomes, and obviously this double-stranded DNA genome is transcribed into messenger RNA, and as you see here, and then the messenger RNA is translated into proteins. And so these viruses tend to use much of the machinery that we provide. The group 2 viruses are single-stranded plus stranded DNA viruses. And so the first thing that they have to do is make the complementary minus strand. So they have a transient double-stranded DNA intermediate. And then this guy can then be used to encode messenger RNA. And then the messenger RNA can be translated into proteins. The group 3 viruses are double-stranded RNA viruses, which is pretty unusual. Double-stranded RNA is not a super stable molecule, but there are viruses that have this genome. And so again, these guys have, after strand separation, the minus strand can be used as a template to make more plus strands. And again, I didn't mention this, but I'll back up and say that messenger RNA is a plus strand of RNA. Any RNA that has the necessary ribosome binding sites to facilitate translation is considered a plus strand of RNA. Group 4 viruses are a single strand of plus strand RNA. So the coronaviruses, like SARS-CoV-2, would fall into this category. And so these guys have to encode a polymerase of their own to rapidly make minus RNA, and then the minus RNA is a template to make more plus RNA. And then the plus RNA can be translated into various proteins. The group 5s are a single strand of, well, not necessarily a single strand, as you'll see, but are single-stranded RNA viruses with a minus orientation. So again, these minus-stranded viruses have to make a plus strand of messenger RNA that to facilitate translation into proteins. And then the group 6s, they're also plus-stranded RNA viruses, but they're goofier than the group 4s, more sophisticated. In this particular case, these are the retroviruses, and so they have an enzyme called reverse transcriptase, which utilizes their plus RNA genome as a template to make double-stranded DNA. And then the double-stranded DNA can be transcribed into plus messenger RNA. And then ultimately that can be translated into proteins. And then the final group, group 7, these guys are interesting because they have a double-stranded DNA genome, but a little bit of a tail of RNA. And this little RNA tail gives them the ability to basically prime RNA synthesis. And so again, these guys, this primed RNA then facilitates a reverse transcription of the RNA into double-stranded DNA. And then the double-stranded DNA again is transcribed into plus mRNA. So the symmetry of the protein shell or capsid can be helical, as I said. So here's a classical helical arrangement where there's protein subunits that protect the RNA in this particular case and facilitate the appearance of sort of a helical coil. And again, it often looks like an ear of corn under the microscope, electron microscope. And then an icosahedral is a really cool looking adenovirus. And these guys have a very symmetrical appearance with these equilateral triangles that make up this icosahedral capsid. And then you can see the very pronounced virus spikes that come out of this virus that allow it to bind to receptors on tissue. So another component is the presence or absence of an envelope. And so viral capsids can be naked, as you see here, or in this particular case, viral capsids can have an envelope around them, which the virus steals from the susceptible host cell that it infects, and the envelope is a phospholipid bilayer that's derived from the host cell, as I said. Some of the other features that viruses have then are spikes that stick out into the world so that the virus can bind to receptors. And then within that, often they have matrix proteins that are essential for structure, and then capsid proteins, which provide further structure and protection for the nucleoprotein core. Some of the unifying principles of viruses, all genomes are packed inside particles that mediate their transmission. And so again, that's the primary thing, is having some sort of protein surrounding some sort of nucleic acid to facilitate transmission. The viral genome contains the information for initiating and completing an infectious cycle. And that's not always the case. Now, some viruses are defective and don't have all the genes, or some viruses have sort of given up some of their control to the host cell, and so they need to get some of their features from the host cell, and so you'll see some of that down the road. But anyway, the typical infection cycle involves attachment, penetration into the cell, replication, assembly of new virus particles, and then, of course, they have to release somehow. All viruses are able to establish themselves in a host population to ensure their own survival, and if they can't, they go extinct. So here's a bit of some lab-based information. So the cell culture is the most common method for virus propagation. Obviously, you can raise mice or whatever susceptible host animal. Monkeys is commonly used for the more deadly microbes. But obviously, you can imagine how just heartbreaking it is to maintain animal populations and just constantly kill them off with viral infections, especially for monkeys and things like that. So a lot of virology labs rely heavily on propagation in cell culture, and so this is just an idea of what that might look like. So in a real circumstance, here's an intestinal epithelium. Typically, you need something, an epithelial layer fixed to a surface. In this real model, the surface is tissue and blood. And here is our epithelial cells, and then typically that would also include a mucus layer over the surface. So again, a virus like norovirus that's infecting human epithelial cells would have this to deal with, would have to get through this mucus and get to the epithelial cell surface without being pooped out or without being destroyed in trying to transit this viscous mucus that's full of toxic components. So cell cultures are often not particularly realistic, like you see here with the mucus and the monolayer and an immune response under here and all that. Obviously, you don't want anything getting in the way of virus propagation if you're just trying to grow them in the lab. So sometimes we have non-polarized, non-confluent cells. So these sort of rounded up non-confluent cells, they're not touching each other, that are stuck to plastic or glass. Very different from a polarized monolayer. So here's a polarized monolayer. So it's polarized in the sense that the apical surface that you see here has microvilli in this particular example, and then the basal surface is clearly down here, which is fixed to the, in this case, an artificial substrate. So that's polarized. This is non-polarized because there's no up or down or sideways or anything. This is just one flattened, squashed down cell. With a virus has equal opportunity to get into any exposed region. Whereas here, because this is a polarized monolayer, the viruses really only have the opportunity to get into this apical surface here. And typically, flooded over the surface is some artificial medium that gives the polarized or non-polarized, gives the cells some sort of nutrients. One of the ways we propagate bacterial viruses is suspension culture, where cells can be suspended in an artificial medium, and then obviously the viruses can have access to all surfaces of the cell. So and the same thing with bacterial viruses as well. As I said, these could be bacterial cells or animal cells, depending on what you're trying to propagate.